Well, I'm excited to talk to you today about how we can use transferable biology and breeding to contribute to improving our food systems and climate change. Uh, I'll be giving the introduction of this, but my team will also be highlighting themselves some of the key research they're doing in these areas. So let's get started. The work's been supported by the USDA and the National Science Foundation. And we're also going to go through this research using an approach called Go High. It's goal-directed, hypothesis-driven workflow for applied science that Jeff Morris has developed. And so I hope you appreciate this type of design of the talk. So when I look back over the last century, we've had some tremendous successes in breeding. For example, Norman Borlaug in the Green Revolution has dramatically reduced the proportion of the globe's population with insufficient calories or protein. But we still have a big problem. As you look towards the rest of the century, the vast majority of the people could end up with imbalanced diets that have poor nutrition. So how do we change that? And so I think the vision we have towards a healthy globe looks a lot more like this, where we have improved adapted row crops, alternative proteins, so that everybody has enough calories and protein. And then we also have bred fruits and vegetables and microbial foods that help deal with imbalanced diets. And of course, the other giant goal we need to address is climate change, whether that's adapting our crops to their new environments, but also I think it really means about dealing with carbon sequestration and carbon removal technologies. How can we get our plants to capture 20 gigatons of carbon per year? So what's our lab's mission? It's to accelerate plant breeding to improve global health and carbon sequestration. So why are we working on plants? Well, when you think about those healthy diets, things like fruits and vegetables are really going to be a key player there. And when we think about carbon sequestration, plants essentially occupy over 80% of the globe's biomass, and so they're going to be key for carbon sequestration. So currently, uh, genomic selection is being effectively applied to about 10 species. But I think we're moving into a period where we really need to get genomic selection to be applied to perhaps over 1,000 species. And so fundamentally, that's the question. How do we accelerate it for so many species? Part of that is essentially building the tools so that breeders can just combine genomics with information technologies. And for example, in the USDA, we're trying to get those set of tools through a project called Breeding Insight into the hands of specialty breeders. Genome-wide prediction works well, but it has limitations. Within population, accuracies in a major crop species can be very high. Between populations can drop off substantially. And then if you're working in a specialty species with smaller training sets, additionally, those accuracies are low. And essentially between species, there's no accuracy. So the question our group is asking is, can we add in the rules of life to help constrain some of these genome-wide prediction models so that we'll get a boost in accuracy? And I'll talk the rest of the time about how we're trying to do some of those elements. And so this is where we're asking the question about the rules of life. And essentially, how can we use the information that we're learning about all sorts of model systems and applying them across this much wider set of species? And I don't think it's really about discovering the rules of life. I think we know a lot of the rules of life. I think it's much more about parameterizing the functions of life. And that's what our group has really been kind of focused on, is how to do that. And so what I'll be talking about today are essentially some of the hypotheses we have on the transferability of these rules of life across a wider range of species. And I'll tell you about where we are with transferring these parameterized models in a couple of different areas. I also just want to provide a technical reminder that essentially the signal of evolution can be so strong that it's critical that those covariances be considered. Hai Wang and Jacob Washburn in the group produce a really nice set of approaches that are very useful for dealing with these evolutionary approaches when you do machine learning. So the first hypothesis I want to discuss on transferability, and Sarah Jensen will go into detail on this, is essentially the biophysics of protein and RNA structures should be similar and shared essentially across all of life, whether those be bacteria, archaea, or eukaryotes. And that's essentially talking about transferring information over about a 3 billion year period. So I'll let Sarah take it over from here.
Hi, I'm Sarah Jensen, and I'm looking at questions related to transferability across large evolutionary timescales. So we're hypothesizing that biophysics is consistent and transferable across archaea, bacteria, and eukaryotes. And we're using things associated with temperature as our phenotypes because we know that molecular biology is affected by temperature. And so now we're trying to ask questions related to the central dogma to see which elements of the central dogma are transferable, and can we even transfer information across such a large evolutionary time period? So I started working on this project with Emery Chimen, and we hypothesized that prokaryote tRNAs would have a consistent evolutionary response to temperature, and we predicted that machine learning models would be able to pick up on those elements to predict optimal temperature. So we built a model using tRNA sequences to predict prokaryote optimal growth temperature, and what we found was that we were able to build a model that predicted OGT, optimal temperature, with high accuracy across archaea and bacteria, even though we were only using about 0.1% of the total genome space. And the success of these models tells us a few things. So first, it confirms our hypothesis that we can transfer information across domains of life. Second, it tells us that machine learning models can capture key elements of the genome, so in this case, tRNA stability and sequence, to help us learn about molecular evolution. And then third, these models also provide insights into relevant biochemistry that we can follow up on later. So for example, with this study, we found that our models focused on the anticodon and T arms of the tRNA, suggesting that those are the regions of the tRNA that are most important for stability. And so now, having shown that we can transfer information across prokaryotes, I've moved on to looking at proteins. And because we started with that tRNA model, we can get an estimate of optimal growth temperature for any prokaryote species that has a genome, and that estimate is completely independent of the proteins within the organism. And as with the tRNA story, my hypothesis here is that proteins have a consistent evolutionary response to temperature, regardless of where they've come from. And essentially, I run a GWAS on PFAM domains within my prokaryote dataset. And what I find is that there are large portions of the genome and the proteome that seem to be associated with temperature, and that overall about 15% of residues within PFAM domains are associated with temperature in both archaea and in bacteria. And what's even more interesting is that we can actually narrow down to these shared significant sites and look at their physical and chemical properties to try and understand why they're important. So now that we know we can use GWAS to learn about protein domains across prokaryote species, I'm working to apply the same set of analyses to eukaryotic proteins and to find out how much of this is transferable to plants.
So Sarah provides some insights on transferability at the protein level, and we're really hoping to see if there's applications to temperature tolerance in plant proteins. But now I'd like to turn our attention to gene regulation. And of course, many of the key elements of gene regulation are shared across eukaryotes. And by the time we get to plants, only about 150 million years ago, a lot of, nearly all of those transcription families are shared. So what can we transfer at this level?
Hi, I am Caterina Mejia Guerra. When I started working in the testing of these hypotheses, I wanted to know if I could develop accurate machine learning models to transfer expensive biochemical characterization to determine regulatory regions between maize lines. And it worked. We learned to distinguish open chromatin regions from random regions in the genome with a great degree of accuracy. After this initial exploration, we collaborated with Celine Cheung groups in University of Hong Kong to study the in vivo binding preference of 104 transcription factors expressed in the maize leaf. In this project, we found that sequence alone allows the training models to have a degree of accuracy of more than 70% for each of the TF, and that models that consider the binding of several TF simultaneously are even more accurate. From building accurate models, we were able to use the K-mer weights to summarize the TF relationships based on their sequence preference. And here we learn that binding preference agrees with TF protein families. And as we know protein families are conserved all the way even to Arabidopsis, we figured out the sequence binding preference might be also conserved. So when we look at the motif level, we found that 61% of the mice TF in our collection bind to similar sequence than the Arabidopsis one, all of which implicates a strong constraint over more than 150 million years in the regulatory grammar.
Hi, I'm Travis Reitzman, and I was interested to see if the Kamer models that Catherine developed were directly transferable between maize and Arabidopsis. I evaluated Kamer models trained with Arabidopsis RD26 binding data on a holdout set of Arabidopsis data, shown in black, and then on binding data from an orthologous TF in maize, shown in blue. When I trained a model on a mixture of data from both species, shown in green, I saw that it had almost equivalent precision and recall to the within-species Arabidopsis model, despite very different genomic contexts across the two species. So suspecting that transcription factor binding was transferable across Arabidopsis and maize, and knowing that TFs bind to open chromatin, a natural next step was to see if chromatin accessibility was transferable across angiosperms. I trained a convolutional neural network to predict chromatin accessibility using ATAC-seq from 13 angiosperm species. Generally, this panangiosperm model shows high precision and recall on test sets across many of the species. However, I'm still looking into whether the harder to predict species are due to poorer data quality or actual biological mechanisms. To roughly test if expression models were transferable, I took maize-trained tissue-specific models that predict mRNA levels using sequences around the transcription start and termination sites and tested those against Arabidopsis gene sequences. Without looking at any Arabidopsis sequence, these maize models are still able to capture some of the variation in expression levels due to nearby cis-regulatory sequence. The key question that we are still following up on is, how much better can these models be if trained across species?
So I think Catherine and Travis have shown that there is good transferability across plants, and this will only improve over time. While they didn't show it today, these models can have single base pair sensitivity, which is really useful for genomic selection. In the future, we really need to explore the question about how far we can leverage these models, if we can include models from animal and fungi. We do know that about half of the TF families are shared with them, so possibly. Our next area has been whether we can connect the mutations that likely affect gene activity to phenotype. Unfortunately, because of lack of information, most of our work has really been on disruption of genes or proteins. We, like many groups, have successfully been able to use evolutionary constraint approaches like GERP, but we also saw some weaknesses as we go across species with very different physiology. Guillaume's going to talk about some of the ways he's used these types of approaches to improve upon those standard approaches.
Hi, I'm Guillaume Ramstein. I am one of the scientists in the Bucker lab who addressed the general hypothesis that putative deleterious mutations decrease fitness in plant species like sorghum, cassava, or maize. To address this hypothesis, we tested whether accounting for putative deleterious mutations increased the accuracy of genomic predictions for traits that are related to fitness, like plant height or grain yield. We determined putative deleteriousness of variants by GERP scores, which quantify conservation of variants across species. In quantitative genomic studies, we determined whether information from GERP scores could improve prediction accuracy. In general, our results indicated small to moderate improvements in prediction accuracy by accounting for putative deleteriousness. In hybrid maize, we recently assessed genomic prediction in cross-population validations, where we trained models in one panel and validated them in a distinct panel. Accounting for GERP scores did not improve prediction accuracy for plant height, despite an apparent enrichment of genic regions for causal variants. But in some other cases, for grain yield for example, accounting for putative deleteriousness did improve prediction accuracy, even after accounting for enrichment of genic regions for causal variants. To investigate further the functional relevance of deleterious mutations, we made the hypothesis that fitness effects are consistent across plant species. To address this hypothesis, we first modeled the relationship between genome sequence variation and fitness effects of CYP mutations through genomic annotations. using sequence analysis based on bioinformatic methods or deep neural networks, we derived annotations about genomic structure and protein function at various scales, either contiguous genomic regions or single DNA sites. Then, we used evolutionary constraints across 27 species, not including maize, as a proxy for fitness effects of SNPs. And finally, we used a typical machine learning technique, random forest, to learn the relationship between our genomic annotations and evolutionary constraints. The model used to predict evolutionary constraints allowed us to rank variants with respect to putative fitness effects. To determine whether these ranks were meaningful, we tested a set of expectations which followed from our hypothesis. Our first expectation was a negative relationship between evolutionary constraints and within species variability, as reflected here by minor allele frequency, MAF. This expectation was supported, especially for predicted evolutionary constraints, compared to observed evolutionary constraints. Our second expectation was an improvement of genomic predictions by evolutionary constraints in cross-population validation. Here, as a baseline, I'm showing the increase in genomic prediction accuracy achieved by random weights for prioritizing SNPs in genomic prediction of grain yield in hybrid maize. In this case, using predicted evolutionary constraints as weights to prioritize SNPs resulted in a significant increase in prediction accuracy compared to the random weights. These results, along with the negative relationship between evolutionary constraints and minor allele frequency, suggested that fitness effects learned from conservation across plant species are functionally relevant.
So you heard from Guillaume that we're making lots of progress in this area, but we're still concerned that we have maybe too much diversity in physiology, especially for whole plant traits like yield. So our hypothesis is that if we use the information from the Anthropogony, which have a shared physiology, we'll be able to better estimate some of these fitness drops. So to test this, we're working with Toby Kellogg and Matt Hufford, Jeff Rossabara, Adam Siepel, and Chi Sun. Together, we're going to try to look at nearly 900 species that are closely related to maize and sorghum. And they share this key shared physiology, and together, their evolutionary branches span about 1.5 billion years. So one way to think about this is that we have a saturation evolutionary analysis of a consistent physiology. Every ancestral base has been mutated about 50 times across these samples, and we're hoping that effectively we can measure constraint directly while conditioning on dosage.
Hi, I'm Boxing. I'm interested in how to use the evolution knowledge learned from interspecies omics data to understand the intraspecies natural relation. Using the Anthropogony trip, we proposed the hypothesis disruption in Anthropogony shared physiology reduce misfitness. We started this by identifying shared functional genomic elements. We aligned the genome sequence of each Anthropogony species against the maize no-coding sequence, and identified the conserved no-coding sequence across the Anthropogony trip. A very large proportion of those conserved no-coding sequences could be explained by maize regulatory elements, and we predict disruption of CNS result into gene expression dysregulation. Calculating measured the gene expression across 7 tissues for about 300 maize inbreeding lines, and the genome sequencing data of those maize lines have been released. Using the genome sequencing data, we could identify the disruption of CNS in this maize population. By comparing CNS disruption with gene expression level, we found the loss of CNS is associated with extremely low gene expression.
Hi, I'm Joe Gage. Similar to Guillaume's work estimating fitness consequences of non-synonymous SNPs, and Baoxing's work studying how mutations in conserved non-coding sequences affect RNA expression, I'm interested in how rare, putatively deleterious variants affect protein expression. Building off of previous work by Kremling et al., who showed that rare variants are associated with abnormal RNA expression, we hypothesized that the effects of rare variants should also be visible at the protein level. To test this, we predicted that genes with rare alleles should show higher variability in protein abundance due to abnormal upregulation or downregulation. Using proteomic data from nearly 100 inbred lines and dense SNP genotype data, we found that the distribution of protein abundance for genes with rare alleles in their promoters, UTRs, and exons, shown by the red distributions here, is more variable than for genes without any rare variants, which are shown in gray. This pattern is particularly pronounced for variants in the 5' UTR, and currently we are working to understand just how rare variants cause this observed pattern in protein abundance.
So I think we're moving forward to completing some of the elements of the central dogma, but how do we make better cross-population predictions now? Our prior work in large biparental populations had suggested that maize harbors lots of shared QTLs for component traits, but there were also a large allelic series. Merritt will show with much more high resolution that we have confirmation of that. And then Anju will show about how we're trying to use expression and expression haplotypes as a shortcut for this now.
Hi, I'm Merritt Birch. In this project, we hypothesize that shared phenotypes across populations are the product of underlying shared loci. To address this hypothesis, we've been looking at the correlation of GWAS results from 4,500 different traits between 5,000 half-SIB lines against 282 diverse inbred lines. There's very little correlation in transferability of GWAS results at the SNP level, although this relationship is highly significant. These results suggest that a largely non-overlapping allele series contributes to these phenotypes between populations. Moving our results from SNPs to a higher genetic level like genes or haplotypes could increase this transferability, especially in populations where richer allele series contribute.
Hello everyone, I am Anju Giri. I'm working on questions related to transferability across population. We are working on the hypothesis that cis expression summarizes rich allelic series and reduces dimensionality over SNPs, which helps to improve transferability across population. For that, first we get mRNA labels produced by cis regulatory regions, focusing on the regions right around the genes and using that cis estimates of expression in prediction of complex traits across two diverse populations in maize. So in order to get cis estimates of haplotype expression, we use mixed model approach using the expression data that were previously collected by Kremling et al. On 7 diverse tissues, and haplotype relationship metrics in the gene region is cis, and we combine all the cis haplotype relationship metrics and model them as trans effect in our model. So looking at the results here, we first look at the correlation distribution between measured and haplotype imputed expression across all genes. If we just look at the correlation within each tissue, the haplotype expression is modestly correlated with a mean of around 0.44. But looking across the tissues, using just the haplotype expression, we see stronger correlation of genes across the tissues compared to the measured expression, which is this cyan peak here in right plot. So partitioning out the trans component that includes more developmental and environmental noise in expression, we get higher transferability of expression across these diverse tissues here. So using this cis haplotype expression, we look at the genomic prediction model to predict 26 complex traits in maize. When the model was trained in small diverse population of 280 individuals, we see consistent increase in accuracy over haplotypes. However, the increase in accuracy was larger when the model was trained in NAM, the population of 5,000 individuals from 25 biparental families. So the higher population size and less diverse haplotypes in NAM seems to be helping here in this higher increase in accuracy when the model was trained in NAM and tested in Goodman panel, which is a diverse panel of 280 individuals only. So when we get down to mRNA level and break down expression to cis haplotype that summarizes allelic richness, we are seeing some transferability here.
So where are we on transferability? Well, we've certainly not completed the loop. But I think we're getting to a point where, for example, when it comes to protein structure and activity, we're seeing transferability across the main domains of life. When it comes to regulatory grammars, I think we're seeing decent transferability across plants, about 150 million years. When we're looking at deleterious mutations, we're learning useful things all the way across the grasses, or about 70 million years. And I think that's going to improve a lot when we focus in on the species with just the similar physiology of the Anthropogoniae. And we're seeing that within the Anthropogoniae, that 18 million year divergence. The regulatory elements that are distal, we're getting really useful information on when things are broken there. If we actually want to improve the predictions we have today, then we have to use things like expression, and those cis-estimated RNA haplotype effects really do seem to be transferable between maize varieties, anywhere from the order of 10 to 1,000 years. But I want to highlight that nobody should really be driving a whole breeding program based on any of these data yet. Of course, using standard genomic selection approaches are the way to go. But I think we can see that perhaps over the next 5 years, we're going to be able to help close the circle and really produce and provide a value add. And I think that becomes really important when you think about the fact that sequencing a genome is going to drop to about $2,500 a genome, and perhaps 1 million or more species will be sequenced in the next decade, for example, by the Earth Biogenome Project. But to functionally parameterize these genomes costs in the order of anywhere from $5 to $100 billion. So even though I think we've made progress and there's still a ways to go, I think pushing on an improved transferability is a really useful and key goal. So while 20th century success in breeding looks something like this, I think we're moving to a phase where essentially 21st century success in breeding really is about combining the knowledge across multiple domains, and by doing that, I hope we can tackle a couple of these really big issues facing society with really diverse teams. And so I'll end there. You've heard from some of the great people in my group who did the research, and I just want to highlight that Sarah Miller, Cynthia May, and Peter Bradbury are group leaders who really help the whole team work together and with all of our great collaborators. I'd be happy to take some questions.